Usually, medical diagnostics rely on, a sense of visual finality.
Right.
Like you break your arm, the X-ray reveals the jagged line, and the doctor just points right to the fracture. The problem is visible, the solution is mechanical, and, well, the whole process feels grounded in a reality you can actually see.
Yeah, I mean, we've always relied on visual confirmation in medicine. It gives us this sense of certainty when we can categorize a pathology just by looking at a scan or a slide under a microscope.
Exactly. But that certainty completely vanishes when you step into the world of next-generation
Oh,
therapeutics.
absolutely.
Because we're looking at treatments for neurodevelopmental disorders or targeted cancer therapies, where the drug itself is entirely invisible to the human eye. The whole landscape has shifted away from synthetic chemicals and into the realm of these microscopic biological vessels.
Right. We've reached this point where the mechanism saving your life is, frankly, vanishingly small.
Mhm.
Traditional visual confirmation is just no longer physically possible. I mean, these therapeutics exist below the diffraction limit of visible light.
Which is wild. And that's exactly what today's deep dive is focused on. How do manufacturers navigate that invisible reality? We've got a really dense stack of sources today to help us figure this out. We are pulling from scientific articles, manufacturer specifications from companies like Malvern Panalytical, Hyperion Analytical, and Particle Metrics, plus a bunch of regulatory
That's
guidelines.
a lot of material to get through.
It is, yeah. But our mission for this deep dive is clear. We are bypassing the foundational biology to look directly at the manufacturing floor. Specifically, how quality control scientists use a technology called Nanoparticle Tracking Analysis, or NTA.
NTA is right.
We want to see how NTA verifies the exact size distribution and concentration of exosomes. Basically, how do you mathematically prove to the FDA that an invisible therapeutic is safe, consistent, and actually ready to be injected into a patient?
Yeah, and the stakes in this specific area of chemistry, manufacturing, and controls, which we often just call CMC in the industry, the stakes are uniquely high here.
Mm.
Because exosomes, or extracellular vesicles, they're essentially just biological envelopes. They range from like 30 to 150 nanometers in size.
No.
And cells use them to commute vital cargo, like RNA, lipids, and proteins all throughout the body.
Okay, let's unpack this for a second because their ability to naturally transport complex biological material is like exactly what makes them such revolutionary candidates for drug delivery, right?
Exactly. A pharmaceutical company can theoretically hijack these envelopes, pack them with therapeutic RNA, and direct them to target a very specific tumor.
Wow.
They're also heavily involved in early cancer detection. Like, liquid biopsies are now searching for the specific exosomes a tumor leaves behind in the bloodstream.
But I mean, engineering them in a lab is one thing. Scaling that up into a viable pharmaceutical product has to be a nightmare, regulatory-wise.
Oh, it's a brutal hurdle.
Yeah.
The FDA demands absolute proof of the contents of the vial. I mean, you cannot establish the therapeutic dose, let alone predict its pharmacokinetic behavior, if you don't know the precise concentration and size of your exosomes.
I always think of this manufacturing challenge like trying to accurately count millions of highly specific, invisible dust motes suspended in a blizzard.
That is a great way to put it.
And if you're off, the medicine just doesn't work. The biological fluids used to culture these therapeutics are what chemists call polydisperse, right?
Right, polydisperse. And polydispersity is essentially the enemy of simple analytics. Like, a monodispersed sample would be analogous to a jar of perfectly identical marbles.
Nice and easy to count.
Exactly.
Mm.
But biofluids, on the other hand, they are a chaotic soup, just particles spanning a massive range of sizes. When manufacturers isolate exosomes from cell culture media, or say blood plasma, they inadvertently pull down protein aggregates, cellular debris, lipoproteins, and all of that occupies the exact same 30 to 150 nanometer size range.
So it's all just mixed in together, and you can't just slide a drop of that polydispersed soup under a standard laboratory microscope and ask a technician to start counting.
No, definitely not. The particles are way too small to reflect visible light in a way our eyes can process.
So the solution to seeing the dust motes in the blizzard requires manipulating the physics of light scattering. How does NTA actually do that?
Well, the physical architecture of NTA relies on a highly controlled environment. The technician injects a precisely known microscopic volume of the liquid sample into a specialized flow cell.
Okay.
And then a specifically shaped laser beam is fired directly through that liquid suspension.
But the system isn't trying to capture an image of the exosomes themselves, right?
Right.
From what I read in the sources, as the laser passes through the liquid, the suspended particles scatter the light,
That's right.
and then a high-speed CMOS camera, which is mounted on a microscope objective, captures a video of those flashes of scattered light against a completely dark background.
Precisely. The camera records the movement of those flashes frame by frame. And because these particles are microscopic and they're suspended in a fluid, they are entirely at the mercy of their environment. The solvent molecules, the water, they are in a constant state of thermal motion, continuously bombarding the exosomes from every possible direction.
Which creates Brownian motion. The exosomes jitter and bounce in random directions based on all those collisions.
Exactly. And the physics dictate that smaller, less massive particles will get kicked around rapidly, traveling greater distances between the camera frames. Larger, heavier particles, they'll exhibit a much more sluggish, kind of constrained jitter.
Okay, so the NTA software tracks the independent trajectory of every single glowing dot on the screen simultaneously. It's basically calculating the velocity of that erratic jitter.
Yes. And that velocity is the critical data point required to determine the physical size of the particle.
Mm-hmm.
But extracting that data requires a pretty complex mathematical translation.
Yeah, and this is where the underlying mechanics of the technology present a massive dimensional paradox, at least based on the Hyperion analytical source. Wait, Brownian motion is inherently three-dimensional, right? The exosomes in the flow cell are being bombarded from all sides, moving up, down, left, right, forward, backward.
Right, it's a 3D space.
But the CMOS camera sensor is a flat two-dimensional plane. It only records movement along the X and Y axes. So if the camera completely loses the Z-axis, the depth, how on earth does a 2D video give us an accurate 3D measurement?
It's a great question. Bridging that gap between a two-dimensional observation and a three-dimensional reality requires the Stokes-Einstein equation.
Okay.
This equation is, frankly, the mathematical bedrock of nanoparticle analytics. It defines the relationship between a particle's hydrodynamic diameter and its translational diffusion coefficient.
Okay, but how does it solve the missing dimension?
Well, it factors in environmental constants, specifically the temperature of the liquid and its viscosity.
I want to focus on that. How temperature and viscosity solve the missing Z-axis, because honestly, that is the most counterintuitive part of the whole process.
They establish the environmental guardrails, essentially. Temperature determines the exact amount of thermal energy driving those molecular collisions. Viscosity dictates the exact amount of physical resistance the fluid applies against the particle's movement.
Right.
So when those two environmental parameters are locked and stable, physics dictates that isotropic diffusion, that random thermal motion, is statistically independent across all axes.
Meaning the mechanical movement of the exosome along the x-axis has absolutely no influence on its movement along the z-axis.
Exactly. Because the axes are statistically independent, the average behavior in any one direction will inevitably mirror the average behavior in the others, provided your observation window is long enough.
Ah, I see.
By tracking the X and Y movement over a sufficient number of consecutive frames, the NTA software calculates the two-dimensional diffusion coefficient. And from that data, statistical probability allows the algorithm to accurately derive the true three-dimensional translational diffusion coefficient.
So we aren't guessing the depth. We are relying on the uniform nature of random thermal motion. Like, if a specific particle exhibits sluggish movement in the X and Y dimensions over, say, 30 consecutive frames, statistical mechanics guarantee it's equally sluggish in the unobserved Z dimension.
Exactly. The software takes that derived three-dimensional diffusion coefficient, plugs it back into the Stokes-Einstein equation alongside the known temperature and viscosity, and computes the exact hydrodynamic diameter of that specific particle. And it does this for every individual particle in the field of view down to the nanometer.
That is just mathematically beautiful, but calculating the size handles only half of the regulatory requirement. Establishing a therapeutic dose requires knowing the absolute concentration, right? The manufacturer has to prove exactly how many exosomes are actually in that vial.
Yeah, and generating that concentration metric is basically a function of pure optical geometry.
How so?
Well, the NTA system is calibrated with the precise dimensions of the illuminating laser beam and the exact field of view of the CMOS camera. It knows, down to the picoliter, how much liquid is being analyzed in that specific moment.
Oh, wow. So if the software tracks and counts like 500 individual particles within that tightly defined optical volume, determining the concentration is really just straightforward division.
That's all it is. It extrapolates that count to establish the absolute number of particles per milliliter. This system doesn't even require any external reference standards or complex calibration curves to generate that baseline concentration.
That's incredibly powerful. But, this is where we have to look at the limitations of the technology. NTA provides an absolute count and a mathematically derived size for every individual particle, which is amazing, but it's not the only analytical tool used in these labs, and it's certainly not flawless.
Definitely not flawless.
For example, dynamic light scattering, or DLS, is still ubiquitous in nanoparticle analysis. If NTA tracks individual particles with such precision, I find it fascinating that DLS is still heavily utilized when it relies on a fundamentally different way of processing light.
Well, what's fascinating here is that DLS processes the optical data by generating an intensity-weighted bulk average.
Yeah.
So instead of tracking individual particles one by one, it analyzes the fluctuating intensity of the scattered light across the entire sample simultaneously.
Which, as the sources point out, creates a massive bias toward larger particles. In that polydispersed blizzard we discussed earlier, a 200 nanometer protein aggregate is going to scatter exponentially more light than a tiny 40 nanometer exosome.
Oh, massively more. A sample containing just a few large aggregates will generate an optical signal so overpowering that the DLS sensor essentially goes blind to the millions of smaller exosomes.
Wow!
The algorithm averages the entire signal, pulling the reported size distribution heavily toward the massive junk particles. NTA circumvents this bias entirely by utilizing a number-weighted distribution.
Right, because NTA visually tracks the particles one by one, so a massive, brightly scattering aggregate still only registers as a single data point. Its sheer brightness doesn't mathematically override the tiny, faint exosome bouncing right next to it.
Exactly. But while that individual tracking is a distinct advantage, NTA encounters its own severe limitations, especially when analyzing highly complex, unpurified biological samples.
Like raw blood plasma.
Yeah, exactly. When a sample like that contains a high concentration of large, highly refractive lipoproteins, the sheer volume of scattered light actually overwhelms the CMOS sensor.
Oh, it causes a washout effect. According to the cell guidance system's source, the dark background of the video feed basically turns into a bright gray haze.
Yes, and when that optical contrast degrades, the software can no longer distinguish the faint flashes of the smallest particles. The sensitivity threshold drops dramatically. In highly polydispersed environments, NTA frequently fails to detect particles below 60 nanometers.
Wait, 60 nanometers? That blind spot is catastrophic for exosome therapeutics, considering the target size range drops all the way down to 30 nanometers. A manufacturer could be failing to measure the most therapeutically active and abundant fraction of their entire dose. So NTA is brilliant, but it has a blind spot for the tiniest players if the big guys are hogging all the light.
It really is a major issue. Head-to-head analytical studies demonstrate the severity of this limitation. When analyzing complex biofluids, NTA can actually underestimate the true total concentration of extracellular vesicles by an entire order of magnitude like 10x compared to orthogonal technologies like microfluidic resistive pulse sensing or MRPS.
Because MRPS avoids the washout effect entirely by measuring physical volume via electrical resistance as particles pass through a nanopore, it doesn't rely on light scattering at all, so a bright background doesn't corrupt the data.
Exactly. With NTA, if the camera cannot register the flash of light, the particle simply does not exist in the dataset. So manufacturers are forced to implement aggressive purification protocols to strip out those large scattering aggregates before NTA can provide a reliable count of the smallest exosomes.
But even if a manufacturer perfectly purifies the sample and the NTA software flawlessly tracks a 100 nanometer particle, there's still a massive regulatory hurdle. A bouncing dot on a screen is just a bouncing dot. Bright field NTA, the standard light scattering mode, it provides the size and the count, but it gives absolutely zero biochemical data.
Right. And the FDA requires proof of identity. A manufacturer cannot simply claim, we have a billion particles of the correct size. They must guarantee those particles are the specific therapeutic vesicles intended for the patient, and not just perfectly sized droplets of lipid or cellular waste.
So solving that specificity problem requires a significant upgrade to the methodology, which the sources highlight as fluorescence NTA or FNTA.
Yes, FNTA introduces biochemical targeting. Basically, manufacturers tag the sample with fluorophore-conjugated antibodies that are engineered to hunt down and bind exclusively to specific proteins residing on the surface of the exosomes.
And the International Society for Extracellular Vesicles strongly recommends targeting tetraspanins for this, right? Proteins like CD9, CD63, and CD81. They're deeply embedded within the exosome membrane, making them highly reliable biological markers.
Right. They act as a molecular filtering system.
Hmm.
So a fluorescent tag engineered to target CD63 will successfully coat the exosomes, but it will completely ignore any random protein aggregates in the sample that lack that specific surface marker.
Then the technician swaps the standard laser for a specific excitation wavelength, usually around 488 nanometers, and places a fluorescence filter directly in front of the camera sensor. That filter physically blocks all the scattered white light reflecting off the non-target debris.
Leaving the camera to only register the emitted fluorescent light. The background junk vanishes from the video feed entirely, and the software tracks only the true CD63 positive exosomes. It provides this highly specific size and concentration profile of the actual therapeutic.
But okay, let's look at the physical mechanics of this tagging process for a second. We are taking a fragile 50 nanometer exosome and forcefully wrapping it in a layer of bulky antibodies to make it glow. We are physically altering the diameter of the exact particle we are attempting to measure. Doesn't that invalidate the size distribution data required for quality control?
You'd think so, but NTA's extreme sensitivity actually transforms that physical alteration into a critical diagnostic metric.
Wait, really?
Yeah, according to Malvern Panalytical, the tracking algorithm is precise enough to reliably detect size shifts as minute as 3 nanometers when a particle is coated with antibodies or surfactants.
Detecting a 3 nanometer shift is an insane level of optical resolution.
It really is.
Yeah.
And quality control scientists rely on that specific shift to validate their protocols. They analyze the sample in standard bright field mode, first to establish a baseline size, and then they analyze the fluorescently tagged sample. Observing that microscopic shift in the size distribution provides physical, mathematical confirmation that the antibody is successfully bound to the surface of the exosomes. It proves the labeling process worked.
Oh, that's brilliant. It's a feature, not a flaw.
Exactly.
And for labs looking to bypass the complexity of fluorescent labeling entirely, there's interferometric NTA or INTA. It offers a label-free alternative.
Right. INTA from Bruker integrates the standard particle tracking algorithm with advanced optical contrast data. Since lipid-based exosomes have a different refractive index than dense protein aggregates, they scatter light differently, even if they're the exact same size. INTA analyzes those subtle variations to mathematically separate the populations without needing antibodies at all.
So we have unpacked the three-dimensional math, the fluorescent specificity, and the label-free optical contrast. Now, let's transition from the theoretical physics to the rigid demands of a GMP good manufacturing practice facility. Because experimental biology tolerates variability, but a pharmacy shelf demands unyielding standardization.
Absolutely. The transition from the laboratory bench to commercial manufacturing is heavily regulated by lot release quality control protocols. In ISO 5189 accredited clinical environments, every single batch of an exosome therapeutic must undergo rigorous NTA analysis to verify lot-to-lot consistency.
And the MISAV 2023 guidelines actually dictate that any clinical application of extracellular vesicles must include exhaustive measurements for both particle size and concentration. NTA is perfectly suited for that dual mandate, but the statistical thresholds manufacturers impose on the data are intensely strict.
They have to be. When quantifying these therapeutics via NTA, quality control protocols typically mandate a coefficient of variation, or CV, of less than 15%.
Let's dissect why that 15 per CV is the absolute ceiling for a passing grade. In a fluid biological manufacturing system, demanding less than 15% variance from batch to batch requires incredible precision. If a batch drifts to, say, a 25% variance in size or concentration, the manufacturer is actively altering the therapeutic index of the drug.
Right. If the size profile creeps higher, it often indicates the exosomes are aggregating. Injecting aggregated vesicles could trigger a dangerous immune response in the patient rather than delivering the intended genetic cargo. And conversely, a drop in concentration means the patient receives a subtherapeutic dose, rendering the whole treatment ineffective.
To monitor and prevent that drift, QC scientists plot the NTA data across Levy-Jennings charts. These charts track the average size and concentration metrics over a minimum of 30 consecutive manufacturing lots.
The Levy-Jennings chart functions as the manufacturing suite's early warning system. Like, if lot number 34 reveals a size distribution that has crept 10 nanometers higher than the historical baseline, it triggers a critical warning limit.
The batch is immediately quarantined, production is halted, and engineers have to go investigate the bioreactors, the purification columns, and the buffer solutions just to isolate the variable before that compromised medicine ever leaves the facility.
Exactly.
So what does this all mean for you listening at home? It demonstrates how the abstract physics of Brownian motion and scattered light are weaponized to ensure patient safety. If you or a loved one are receiving a next-generation exosome therapy to, I don't know, regenerate cartilage or deliver targeted immune therapy for cancer, the rigorous application of NTA ensures that the millions of invisible vesicles entering your body were individually tracked, counted, verified, and standardized against decades of statistical mathematics. It completely removes the guesswork from the microscopic world.
And if we connect this to the bigger picture, the evolution of NTA from a basic particle tracker to an essential component of GMP manufacturing represents our relentless drive to engineer solutions at the nanoscale. I mean, overcoming the limits of human sight is what pushes medicine forward. We are building tools to exert total control over environments we cannot even perceive.
It's incredible. But before we sign off, I want to leave you with a scenario that current regulatory guidelines are just beginning to grapple with. We've established that the entire mathematical architecture of NTA, the Stokes-Einstein equation, relies on a fundamental assumption about the particles it measures. It assumes they are entirely passive. It assumes they are perfect spheres drifting aimlessly, driven solely by thermal collisions with the solvent.
Right, that passive behavior is the core requirement for calculating diffusion.
But bioengineering is rapidly moving past passive therapeutics. Laboratories are currently developing synthetic, non-spherical exosomes, and they're even engineering active nanobots equipped with self-propulsion mechanisms. They are designing particles that actively swim towards specific cellular targets.
And when a microscopic particle generates its own kinetic energy, it completely shatters the laws of Brownian motion. The random isotropic diffusion that NTA relies on just ceases to exist.
The Stokes-Einstein equation essentially collapses. So, here's the question for you to mull over. How do you measure the diffusion of a therapeutic that refuses to simply diffuse? We have built incredible mathematical models to conquer the passive, invisible world. But when the medicines of the future start swimming on their own, how will analytical chemists and regulatory agencies adapt their tools to measure an active microscopic reality?
This raises an incredibly important question that will definitely define the next decade of nanoparticle analytics and quality control.
It absolutely will. We'll leave you to ponder the physics of swimming nanobots. Thank you for joining us on this deep dive into the invisible machinery keeping our future medicines safe. Keep questioning what you can't see, and we'll catch you on the next one.